Long before anyone had a word for it, the ocean glowed. Sailors in the ancient world would stand at the rail of their ships and watch the water ignite beneath them. Trails of pale blue light streaming from the hull, the wake shimmering as though the sea itself were burning from within. They had no framework to explain it, no chemistry, no biology, no understanding of the microscopic life teeming in every cupful of seawater. What they had were stories. Aristotle, writing in the 4th century before the Common Era, described the phenomenon with the careful eye of a naturalist, noting that certain substances, dead fish, certain types of wood, the slime of particular marine creatures, produced a cold light, a glow without heat that seemed to defy the basic logic of fire. He did not call it bioluminescence. That word would not exist for more than 2,000 years. But he recognized it as something distinct, something fundamentally different from the flames that heated forges and cooked meals and burned cities. The light of the living world, he understood, played by different rules. Pliny the Elder, writing 4 centuries after Aristotle, catalogued the phenomenon with the enthusiastic disorganization that made his natural history both maddening and indispensable. He described the glow of jellyfish, which he called pulmo marinus, the sea lung, and noted that if you rubbed a walking stick with their slime, the stick would glow in the dark like a torch. He described luminous shellfish and the pale radiance of rotting fish scales. He recorded the observation, attributed to earlier sources, that the mouths of people who ate certain luminous sea creatures would glow as they chewed. Pliny was not always a reliable reporter. He blended careful observation with second-hand rumour and outright legend in ways that later scholars spent centuries untangling, but his descriptions of marine bioluminescence are detailed enough that modern biologists can identify the organisms he was likely describing. The jellyfish were almost certainly members of the genus Pelagia, the mauve stingers, whose bioluminescent mucus is well documented today. The glowing shellfish were likely pidox, boring clams of the family Phollididae, whose bioluminescence was still being studied by European naturalists 18 centuries after Pliny first mentioned them. What neither Aristotle nor Pliny could have known is that the phenomenon they were observing, this cold fire, this living light, is one of the most widespread and ancient biological innovations on the planet. Bioluminescence has evolved independently at least 50 separate times across the tree of life, in organisms as different as bacteria and sharks, fungi and fireflies, single-celled dinoflagellates, and deep-sea fish with jaws longer than their bodies. In the ocean, where the phenomenon is most concentrated and most spectacular, an estimated 3/4 of all organisms living below 200 meters are capable of producing their own light. The deep sea is not a place of total darkness. It is a place of manufactured light, a world where every living thing is either a lantern or is trying very hard not to be seen by one. The chemistry behind it is elegant and, in its broadest outlines, remarkably consistent. A light-emitting molecule reacts with an enzyme in the presence of oxygen, and the energy released by that reaction is emitted as a photon, a single packet of visible light. The specific molecules vary enormously from one lineage to another, reflecting those 50-plus independent evolutionary origins. But the basic principle is always the same. A molecule is oxidized. A photon is released. The darkness retreats. one flash at a time. Tonight, we are going to trace the history of this light, not the human history of its discovery, though we will touch on that, but the deeper history, the evolutionary history, the story of how and why the ocean filled itself with living lanterns over hundreds of millions of years. We will begin in the oldest oceans, in the warm, shallow seas of the Precambrian, where the first faint glimmers may have appeared in organisms so simple they had no eyes to see them. We will descend into the deep sea, where bioluminescence became not a novelty, but a necessity, a language spoken in flashes and pulses by creatures that had never seen sunlight and never would. We will meet the animals that use light to hunt, to hide, to seduce, and to scream for help. And we will end in the laboratories and research vessels of the modern era, where scientists are still discovering new forms of bioluminescence faster than they can explain the ones they already know. The ocean has been glowing for a very long time. The least we can do is pay attention. I'd like to take a brief moment to thank you for watching and to say that if you're enjoying this content, I hope you'll subscribe. I'm also curious as to how many hours of sleep you got last night. Please tell me in the comments. Perhaps tonight, after watching this video, you'll find you sleep a little better. And now, on with the story. To understand how bioluminescence began, you have to imagine an ocean without eyes. For the first 3 billion years of life on Earth, roughly, there were no visual organs of any kind, no lenses, no retinas, no photoreceptors capable of forming an image. Photosynthetic bacteria had been harvesting sunlight since at least 3.5 billion years ago, and many single-celled organisms possessed simple proteins that allowed them to detect the presence or absence of light and move toward or away from it. But seeing, the active construction of a visual picture of the world, did not exist. The Precambrian ocean was a world experienced through chemistry, through touch, through the slow diffusion of molecules across membranes. In a world without eyes, bioluminescence could not have served any of the functions we associate with it today. No luring of prey, no startling of predators, no flashing of coded signals to potential mates. If bioluminescence existed in the Precambrian ocean, and many biologists believe it did, it must have evolved for reasons that had nothing to do with being seen. The leading hypothesis is that the earliest bioluminescent reactions were not about producing light at all. They were about neutralizing oxygen. Before the Great Oxidation Event, roughly 2.5 billion years ago, most life was anaerobic. It thrived in the absence of oxygen, and oxygen was toxic to it. As cyanobacteria began flooding the oceans with molecular oxygen as a waste product of photosynthesis, anaerobic organisms faced a crisis. Oxygen damages proteins, disrupts cell membranes, and generates free radicals that can destroy the machinery of a living cell. The bioluminescent reaction, at its core, is an oxygen-consuming reaction. It binds molecular oxygen to a substrate, neutralizing it, and the light that results is a byproduct, an accident of the chemistry involved in detoxifying a poisonous gas. Under this hypothesis, the very first bioluminescent organisms were not trying to glow. They were trying to survive, and the glow was simply what happened when they succeeded. This idea, sometimes called the oxygen detoxification hypothesis, explains why bioluminescence is so ancient and so widespread. If it began as a metabolic defense billions of years ago, it would have been present in many lineages before they diverged, providing raw material that evolution could later repurpose once eyes appeared. It also explains why bioluminescence is found in organisms that share no other obvious connection: bacteria, dinoflagellates, jellyfish, comb jellies, worms, crustaceans, squid, fish, and even a few sharks. The chemical pathways differ dramatically from one group to another, using different substrates and different enzymes, which tells us that most of these lineages did not inherit their bioluminescence from a single common ancestor. The sheer diversity suggests that the underlying chemistry is simple enough and useful enough that it has been independently reinvented dozens of times whenever the right selection pressures arose. The Cambrian explosion, roughly 540 million years ago, changed everything. Animal body plans diversified wildly, and among the most consequential innovations was the image-forming eye. Suddenly, organisms could see each other, and bioluminescence was thrust from metabolic accident onto the evolutionary stage. A flash could startle a predator. A steady glow could attract a mate. A pattern of pulses could identify species in the darkness. In shallow sunlit waters, producing your own light was mostly a way of advertising your location to predators. But in the deep sea, where sunlight could not reach, bioluminescence exploded into the dominant form of communication, and it has remained so ever since. The fossil record tells us almost nothing directly. Light does not fossilize, but molecular clock analyses suggest that bacterial bioluminescence may stretch back billions of years, while the spectacular light shows of deep-sea fish are relatively recent, evolving within the last 100 million years as those lineages colonized the abyss and discovered that making your own light was the most powerful adaptation of all. If you were to take a jar of seawater from almost any ocean on Earth and examine it under a microscope, you would find, among the diatoms and copepods, a scattering of tiny armoured cells, each one roughly the size of a grain of sand, each one equipped with two whip-like flagella that allow it to spin through the water column. These are dinoflagellates, and they are among the most important organisms on the planet. They produce a substantial fraction of Earth's atmospheric oxygen, form the base of countless marine food webs, and are responsible for the toxic algal blooms known as red tides. A significant number of them, particularly members of the genus Noctiluca, are bioluminescent, capable of producing a brilliant blue-green flash when disturbed. Collectively, dinoflagellates are responsible for the most commonly observed form of bioluminescence on Earth. The glowing sea, the shimmering wake, the phosphorescent surf that has been reported by sailors and coastal residents for thousands of years. The mechanism is beautifully simple. Each cell contains tiny structures called scintillons, membrane-bound packets of the light-producing molecules held at a pH that keeps the reaction dormant. When the cell is mechanically disturbed by a wave, a passing fish, the turbulence of a ship's propeller, the shock triggers a rapid change in pH that activates the chemistry. The entire flash lasts roughly 1/10 of a second. A single dinoflagellate produces a flash so faint the human eye can barely detect it. But dinoflagellates bloom in concentrations exceeding tens of millions of cells per litre. And when a wave, or a dolphin, or a kayak paddle disturbs a bloom of that density, millions of cells fire simultaneously, producing the dramatic ghostly blue glow that has inspired poets and terrified sailors for as long as human beings have lived near the sea. Why bother glowing? The answer is elegant and slightly counterintuitive. The flash is a burglar alarm. When a dinoflagellate is disturbed by a small grazer, a copepod say, the flash does not frighten the grazer itself, but it illuminates the grazer, making it visible to the grazer's own predators. A copepod feeding in a cloud of bioluminescent dinoflagellates is suddenly spotlit, and every small fish in the vicinity can see it. The dinoflagellate is calling for help, screaming in a language of light that something is here eating me, and if you eat that something, now is your chance. Laboratory experiments have confirmed that predation rates on copepods increase significantly in the presence of bioluminescent dinoflagellates. The glow does not save the individual cell. That cell is already being eaten. But it saves the population by ensuring that its grazers are themselves consumed more often. Bioluminescent bays, Mosquito Bay in Vieques, the Luminous Lagoon near Falmouth, Jamaica, are ecosystems shaped by the interplay between dinoflagellate density, nutrients, water temperature, and the geography of enclosed coastal waters. These bays are fragile. Mosquito Bay lost much of its glow after Hurricane Maria in 2017, recovering only gradually as conditions restabilized. Other bays have been permanently diminished by development and pollution. Beyond the dinoflagellates, the surface ocean hosts comb jellies, ctenophores, that produce cascading waves of blue and green light along their transparent bodies when disturbed. The iridescent rainbow shimmer people associate with comb jellies is actually not bioluminescence, but the diffraction of ambient light through beating cilia. The true bioluminescence is visible only in darkness. A deeper, steadier glow, generated from within rather than reflected from without. Comb jellies may have diverged from other animals more than 500 million years ago, making their bioluminescence one of the most ancient surviving examples of animal light production, flickering since before the first fish evolved. Below the sunlit surface, below the wind-mixed layer where photosynthesis drives marine productivity, there is a region scientists call the mesopelagic, the twilight zone. It begins at roughly 200 meters and extends to about 1,000, defined by a single physical fact. Sunlight reaches it, but only barely. At 200 meters, on a clear day in clear tropical water, enough blue light filters down for a dark-adapted eye to detect the silhouettes of objects above. Not enough for photosynthesis, not enough for color vision, but enough to create a world where shadows matter enormously. By 500 meters, even that faint illumination has nearly vanished. By 1,000 meters, it has gone entirely. The twilight zone is a world of gradients, a place where light fades from dim to absent across 800 vertical meters, and the organisms that live there have evolved in response to that gradient with a creativity that borders on the surreal. This is where bioluminescence transitions from an occasional phenomenon to a defining feature of the ecosystem, used by most organisms most of the time. The reason is the silhouette problem. In the faint downwelling light, any animal in the water column is visible from below as a dark shape against the brighter water above. Predators exploit this ruthlessly. The evolutionary answer is counter-illumination, bioluminescent organs on the underside of the body that produce light matching the intensity and color of the downwelling sunlight, effectively erasing the animal's silhouette. Hatchetfish, small, deep-bodied fish with rows of photophores along their bellies, can adjust their bioluminescent output in real time. If a cloud dims the surface light, the hatchetfish dims its photophores within seconds. If the fish tilts, it compensates by adjusting individual photophores. The light it produces is blue-green, closely matching the wavelength of residual sunlight at depth. A predator looking up at a counter-illuminating hatchetfish sees nothing, just water, just the normal faint glow of the deep blue overhead. Counter-illumination has evolved independently in dozens of lineages, including squid, shrimp, and sharks. The firefly squid of the Western Pacific, Wheatosenia scintillans, possesses photophores on its ventral surface, tuned specifically for counter-illumination, along with additional photophores on its arms that serve entirely different functions, attracting mates and startling predators. The cookie-cutter shark uses counter-illumination with a sinister twist. Its entire underside glows except for a small dark patch near its throat. Viewed from below, that patch, the only non-luminous part of its silhouette, resembles a small fish swimming alone in the open water. Larger predators approach what they think is a meal and find themselves bitten instead. The cookie-cutter shark has weaponized camouflage, turning a defensive strategy into an offensive lure with nothing more than a patch of darkness in a field of light. The Twilight Zone also hosts the greatest migration on Earth, the Deal vertical migration, in which vast numbers of organisms rise toward the surface at dusk to feed, then descend at dawn to avoid predators. This migration involves an estimated 10 billion tons of biomass moving vertically every day, so immense it registers on sonar as a false bottom, a phantom seafloor that rises and falls with the cycle of day and night. Bioluminescence is woven through every stage. Ascending animals use counter-illumination during transit. Predators use luminous lures and searchlights, and small crustaceans that feed at the surface carry bioluminescent dinoflagellates in their guts. When chased, the disturbed dinoflagellates flash through the translucent walls of their predators' stomachs, potentially betraying the predator's position to even larger hunters. The entire water column, from surface to abyss, is cross-wired with signals of light and darkness, operating on a planetary scale every night for hundreds of millions of years. Below the Twilight Zone, below the last faint echo of sunlight, the ocean becomes something else entirely. This is the bathypelagic, the midnight zone, beginning at roughly 1,000 meters and extending to 4,000 meters or more, a realm of permanent, absolute darkness where the temperature hovers just above freezing. The pressure would crush an unprotected human body in an instant, and food arrives not as a steady supply, but as a slow, intermittent rain of organic debris from the surface. Biologists call this marine snow. Dead plankton, fecal pellets, shed mucus, the occasional larger carcass, all sinking through thousands of meters of black water. Life in the midnight zone is defined by scarcity. Energy is rare. Encounters with other organisms are rare. And in this world of rarity and darkness, bioluminescence is not a supplement to vision. It is the only light there is. The anglerfish is the most famous resident of this world, though fame has not always brought accuracy. The deep-sea anglerfishes, More than 200 species in the suborder Ceratioidae are among the most alien creatures on Earth. Globular bodies, enormous gaping mouths lined with needle-like teeth, and in the females, a modified dorsal spine extending forward over the head like a fishing rod, tipped with a fleshy, glowing lure called the esker. The esker produces light using symbiotic bacteria housed in a specialized chamber richly supplied with blood vessels that deliver oxygen and nutrients from the fish's own circulation. The anglerfish does not generate the light itself. It has domesticated a microbe, housing it in a bespoke organ evolved for no other purpose, and in return, the microbe provides a lure that draws prey directly into the fish's cavernous mouth. The relationship is obligate. The bacteria cannot survive outside the light organ, and the anglerfish cannot glow without them. It is one of the most intimate partnerships in the animal kingdom. But the anglerfish is only one voice in a vast chorus. The dragonfish family, Stomidae, includes some of the ocean's most sophisticated, luminous predators. Most deep-sea bioluminescence is blue or blue-green. These wavelengths travel farthest in seawater and are what most deep-sea eyes can detect. But the loose-jaw dragonfish, Malacosteus, has evolved the ability to produce far-red light at wavelengths most deep-sea organisms cannot see. It has equipped itself with an invisible searchlight, illuminating prey that remain oblivious because their eyes are blind to red. The red light is generated by a fluorescent filter that absorbs the blue-green of the underlying bioluminescent reaction and re-emits it as red. A sniper scope built from chemistry and refined over millions of years of natural selection. The deep sea also hosts organisms whose luminous displays are among the most spectacular in nature. Siphonophores, colonial animals related to jellyfish, can exceed 40 meters in length, their translucent bodies producing cascading waves of blue light like a living marquee. The deep-sea siphonophore Arena uses bioluminescent lures that glow red to attract fish drawn to the unusual color. Tomopteris, a genus of deep-sea polychaete worms, produces yellow bioluminescence, one of the only known examples of yellow light in the deep ocean, through a mechanism scientists have not yet characterized. And the vampire squid, despite its melodramatic name, a gentle filter feeder, ejects clouds of bioluminescent mucus to confuse predators, using light instead of ink. Studies using submersibles and sensitive cameras have found that upward of 90% of organisms in some deep-sea environments produce light. The midnight zone is awash in self-made light, an enormous community of living lanterns flashing, pulsing, and streaming through the black water, a visual environment as rich as any coral reef, invisible to us only because we lacked the technology to witness it. Bioluminescence is often described as a tool, like a flashlight, a lure, or a camouflage device, and all of these descriptions are accurate as far as they go. But they understate something fundamental about the role of living light in the ocean. For many organisms, bioluminescence is not merely a tool. It is a language, a structured system of signals and responses that allows animals to communicate across the vast, dark spaces of the deep sea with a precision and specificity that rivals any terrestrial signaling system. The patterns of light, their color, their duration, their rhythm, their spatial arrangement on the body carry information, and the animals that receive these signals are equipped with sensory systems exquisitely tuned to decode them. The ostracods offer one of the clearest examples. These tiny crustaceans, no larger than a sesame seed, enclosed in a bivalved shell that makes them look like miniature clams with legs, perform bioluminescent mating displays in Caribbean waters that are among the most precisely choreographed behaviors in the animal kingdom. At dusk, male ostracods rise from the reef or seagrass bed and begin secreting tiny packets of luminescent mucus into the water, each packet glowing blue for a few seconds before fading. The male swims upward in a species-specific pattern: a rising helix, a zigzag, a straight vertical ascent, depositing glowing packets at regular intervals, creating a three-dimensional trail of light in the water column. Each species has its own unique display: a specific number of pulses, a specific interval between pulses, a specific trajectory through the water. Females recognize their species pattern and swim toward it. Males of a different species, producing a different pattern, are ignored entirely. The system is functionally identical to the flash patterns of fireflies on land, evolved completely independently. Convergent evolution suggesting that coded light signals are one of the best solutions to the problem of finding a mate in the dark. The language of bioluminescence extends far beyond mating. Deep-sea squid in the family Enoploteuthyidae possess complex photophore arrays, dozens or hundreds of individually controllable light organs of different sizes, colours and positions, capable of producing an extraordinary range of patterns. Researchers have compared these arrays to the chromatophore displays of shallow water cephalopods, which communicate aggression, submission, and courtship readiness through rapid changes in skin color and pattern. The deep-sea squid, living where ambient light is absent, may have evolved an analogous vocabulary written in points of self-generated light rather than patches of pigment, readable only by other squid with the right eyes and neural wiring to interpret the patterns. The evolution of bioluminescent communication has driven the evolution of the eyes that perceive it. Many deep-sea fish have tubular eyes, biological telescopes that sacrifice peripheral vision for extreme sensitivity to faint flashes directly ahead. Some species have multiple retinas in a single eye, each tuned to a different wavelength. The cockeyed squid, Histiotuthis heteropsis, has one enormous eye pointing upward to scan for silhouettes against residual surface light, and one smaller eye pointing downward to detect bioluminescent flashes below. The two eyes solve completely different visual problems, reflecting the duality of the deep-sea visual world. Upward, the last remnants of the sun; downward, the manufactured light of living things. The sophistication of these eyes tells us the bioluminescent environment is information-rich, that the signals exchanged are complex enough to reward increasingly refined perception, and that the arms race between signal and receiver has been running for a very long time. The deep ocean is full of conversations we cannot yet hear, conducted in a light we can barely see, between animals whose social lives remain almost entirely unknown to us. For centuries, the light of the living world was a mystery wrapped in a chemical enigma. Naturalists could observe and describe it, catalogue its appearances across dozens of species, but they could not explain it. Sailors smeared luminous jellyfish slime on instruments to read dials at night, and Robert Boyle, the father of modern chemistry, demonstrated in the 1660s that bioluminescence required air by placing a glowing piece of wood in a vacuum chamber and watching the light die. When he readmitted the air, the glow returned, proving that something in the atmosphere was essential to the reaction, though Boyle had no way of knowing that the something was oxygen. For 2 more centuries, the molecular mechanism remained completely opaque. The breakthrough came from a French pharmacologist named Raphael Dubois, who in 1885 performed a deceptively simple experiment using the bioluminescent clam Pholus dactylus, one of the same Piddox Pliny had described 2 millennia earlier. Dubois ground up the clam's luminescent tissue and divided the resulting glowing slurry into 2 portions. The first he plunged into hot water, which destroyed the light immediately. The second he left at room temperature, where it faded gradually on its own. When he mixed the 2 dead fractions back together, the light returned. Something heat-sensitive in one portion was combining with something heat-stable in the other. Dubois named the heat-stable component luciferin, from the Latin for light-bearer, and the heat-sensitive component luciferase, identifying it as an enzyme, a catalyst that facilitates the light-producing reaction without being consumed. With this single experiment, Dubois established the fundamental 2-component framework that underlies all bioluminescent chemistry: a substrate and an enzyme combining in the presence of oxygen to produce light. The molecular details took another century to resolve, and they proved far more varied and surprising than Dubois could have anticipated. There is no single luciferin. At least 11 chemically distinct versions are known, each associated with different lineages. Dinoflagellate luciferin is a tetrapyrrole related to chlorophyll, an echo of photosynthetic ancestry. The luciferin used by ostracods, called vargolin, is so stable that Japanese soldiers in the Second World War collected dried ostracods and read maps by the blue glow of a moistened palmful. The most widespread marine luciferin, cholinterazine, has been found in organisms spanning 9 phyla, many of which do not produce it themselves but acquire it through diet, passing it up the food chain like a currency of light. Cholinterazine is the luciferin behind one of the most consequential discoveries in molecular biology. In the early 1960s, Osamu Shimomura began studying the crystal jellyfish Aquaria victoria at the Friday Harbor Laboratories in Washington State. He found that the jellyfish used a calcium-dependent protein, which he named aquarin, that produced blue light. But the jellyfish glowed green. Something was converting the blue to green. That something was green fluorescent protein, GFP. For 30 years, it remained a curiosity. Then, in 1992, Douglas Prasher cloned the GFP gene, and Martin Chalfie soon demonstrated it could be expressed in other organisms. GFP could be attached to any protein in any cell, making that protein visible under ultraviolet light in real time. Biologists could suddenly watch proteins move through living cells, track gene expression in embryos, map neural circuits, and visualize tumors. GFP and its colored derivatives became the most widely used tools in modern cell biology. In 2008, Shimomura, Chalfie, and Roger Tsien shared the Nobel Prize in Chemistry, an honor rooted in the cold green light of a creature that had been glowing for 500 million years without anyone thinking to ask why. The deep sea dominates any discussion of marine bioluminescence, and for good reason. It is the kingdom of living light the place where it reaches its greatest diversity and ecological significance. But the deep is not the only part of the ocean that glows. Shallow coastal waters, coral reefs, and the open surface host luminous organisms that are no less remarkable for being more accessible, and in some cases, they are only now being discovered. The assumption that bioluminescence is primarily a deep-sea phenomenon has caused researchers to overlook luminous life in habitats where sunlight is abundant, and the need for self-generated light seems less obvious. Biofluorescence, absorbing light at one wavelength and re-emitting it at another, was long considered a minor curiosity in shallow water marine organisms. Then, in 2014, a research team led by David Gruber and John Sparks published a sweeping survey revealing it to be extraordinarily widespread among marine fish. Using blue light-only illumination and yellow barrier filters that blocked reflected blue light while transmitting the longer fluorescent wavelengths, they photographed more than 180 species fluorescing in vivid greens, reds, and oranges: eels, gobies, scorpionfish, flatfish, sharks, and rays. The patterns were often invisible under normal white light, appearing only when the fish were illuminated with the narrow blue wavelengths that dominate the ambient light at reef depths below about 10 meters. The discovery suggested that a vast visual world exists on coral reefs that human eyes cannot access without special equipment. A world of fluorescent signals and patterns visible to the fish themselves, whose visual systems are tuned to detect exactly these wavelengths, but invisible to us because our eyes lack the filters to separate the fluorescent emission from the ambient blue. True bioluminescence, light generated chemically without any external light source, also occurs in shallower waters, and some of its most intriguing practitioners are sharks. The kitefin shark, Delatius leacher, which inhabits the mesopelagic but regularly ascends to shallower depths, was confirmed in 2021 to be the largest known bioluminescent vertebrate, reaching lengths of nearly 2 meters. Its entire ventral surface is covered with photophores, producing a slow blue glow for counter-illumination in the dimly lit upper reaches of its habitat. The velvet belly lantern shark, a small species found in the eastern Atlantic and Mediterranean, has evolved glowing photophores on its dorsal fin spines, a bioluminescent warning signal advertising the presence of sharp defenses to potential predators, the deep water equivalent of the bright colors used by poison dart frogs and wasps on land. This would represent a bioluminescent form of aposematic signaling, and it works precisely because in the dim waters where these sharks live, reflected color is useless. You cannot warn with a color no one can see. You can warn with a light you make yourself. Coral reefs host their own bioluminescent community, most visible at night. Brittle stars, thin-armed relatives of sea stars, produce green or blue flashes from cells along their arms, and some species shed a flashing, writhing arm as a decoy while they escape into a crevice. Sea pens produce cascading waves of light when touched, the glow travelling from the point of contact outward along the colony over several seconds. Perhaps most unexpectedly, the 2014 fluorescence survey opened a broader question about how much of the ocean's visual environment humans have simply been unable to perceive. The shallows are not merely a well-lit counterpart to the abyss. They are their own kind of light environment, dominated by blue wavelengths, where fluorescence can create private channels of communication invisible to predators that lack the right visual pigments. The boundary between reflected light and generated light is blurrier than anyone suspected, and we have only just begun to learn how to see it. We have travelled from the Precambrian to the present, from the warm surface waters where dinoflagellates flash their burglar alarms to the freezing midnight zone where anglerfish dangle their bacterial lanterns. We have watched the chemistry unfold, substrate and enzyme, oxygen and photon, the same reaction reinvented by evolution dozens of times across hundreds of millions of years. We have seen counter-illumination erase silhouettes in the twilight zone, and far-red searchlights give dragonfish invisible vision. And we have followed a green fluorescent protein from a jellyfish in Puget Sound to a Nobel Prize and a revolution in biology. But the story of bioluminescence is not finished. It is accelerating as new technologies reach parts of the ocean and parts of the genome that were previously inaccessible. The deep ocean remains overwhelmingly unexplored. More than 80% of its floor has never been mapped in detail, and the water column above it, the vast three-dimensional space where most bioluminescent organisms live, is even less well characterized. Every expedition discovers new species, and a significant proportion of them are bioluminescent. In 2020, a remotely operated vehicle off Australia filmed a siphonophore in the genus Apolemia, measuring an estimated 47 meters, potentially the longest animal ever recorded, its bioluminescent body glowing blue in a vast spiral as the ROV disturbed it. Genomic studies are revealing the evolutionary history of light production with unprecedented resolution. By comparing the genes that encode luciferases across different lineages, researchers can now trace which bioluminescent systems share a common ancestor and which evolved independently. A landmark 2019 analysis concluded that bioluminescence evolved independently at least 27 times in ray-finned fishes alone, far more than previously estimated, and that luminous lineages tended to diversify into more species than non-luminous relatives, suggesting that bioluminescence is not just an adaptation but an evolutionary accelerant, a key that opens ecological niches otherwise closed. The practical applications of bioluminescent chemistry have expanded far beyond GFP. Luciferase-based reporter assays are now standard tools in pharmaceutical research, used to screen potential drugs by measuring how they affect gene expression in living cells. Bioluminescent imaging lets researchers track the growth and spread of tumors in living animals in real time without surgery by engineering cancer cells to express luciferase and photographing the faint glow emitted through the animal's body. Environmental scientists are developing bioluminescent biosensors, cells engineered to glow in the presence of specific pollutants or pathogens, providing a rapid and visible indicator of contamination. And synthetic biologists are working on bioluminescent plants, trees and shrubs genetically engineered to produce their own light, which could theoretically serve as self-powered streetlights. The technology is embryonic, but proof-of-concept plants already emit a visible glow. Climate change poses uncertain but real threats to these ecosystems. Rising temperatures and expanding oxygen minimum zones are shifting the distribution of mesopelagic life and altering the timing of the diel vertical migration. Coral bleaching disrupts the reef communities where bioluminescence was only recently discovered. Changes in ocean pH could interfere with the calcium-dependent bioluminescent reactions used by some organisms, and the expansion of coastal dead zones threatens the dinoflagellate populations that sustain the glowing bays humans have marveled at for millennia. The ocean will continue to glow. The chemistry is too ancient, too embedded in marine metabolism to be easily extinguished. But the particular symphony of light that exists today is a product of hundreds of millions of years of evolution, and it is not guaranteed to survive the next century unchanged. Every species lost is a voice removed from the conversation. We are only beginning to read this story, and it would be a particular tragedy to lose chapters before we understand what they say. The ocean has been telling stories in light for longer than any creature has had eyes to read them. What began as a chemical accident, a cell trying to survive the slow poison of oxygen, became, over billions of years, a language spoken by 3/4 of all life in the deep sea. Dinoflagellates flash their alarms in the shallows. Hatchetfish erase their shadows in the twilight zone. Anglerfish dangle their lures in the abyss. Dragonfish hunt in colours their prey cannot see. And a green protein from a jellyfish no one had heard of transformed the whole of modern biology and won a Nobel Prize. We tend to think of the ocean as a dark place, and in a literal sense, most of it is. Sunlight reaches only the thinnest upper layer, and everything below is left to its own devices. But the creatures that live in that darkness did not accept it. They answered it the way life has always answered every challenge the planet has posed, with chemistry, with adaptation, with a stubbornness so old it might as well be called patience. The deep sea is not dark. It is full of living light, and it has been full of living light since before anything alive knew what light was for. Sleep well tonight. Somewhere beneath the waves, the ocean is still glowing, and it will still be glowing when you wake. Thank you for watching.